So for the signal transduction pathway, we've kind of talked about cyclic AMP's role, but there's other things that specifically the 7 transmembrane receptor can do to utilize second messengers. So yeah, we already know about cyclic AMP, but just how there's other hormones and other things that are going to bind to 7 transmembrane receptors, there's other effects that we can have. So the other type of, or I guess subclass of second messengers would be ones that come from PIP2 hydrolysis. So a really long compound known as phosphatidylinositol 4,5-bis, that's where the 2 comes from, phosphate that's what the abbreviated form of PIP2 is, or what PIP2 is an abbreviation of. And this is going to be acted upon by an enzyme known as phospholipase C. Or for short, we just go ahead and call it PLC. And the reason why I'm mentioning this is that phospholipase C is activated by the G-alpha subunit with GTP bound to it. So the G protein that we talked about in the last one that activates adenylate cyclase is also going to play a role in activating phospholipase C. And so when we expose something such as, and I'll switch colors just to really reiterate the point that I'm trying to draw here, such as PIP2 to phospholipase C, we're going to hydrolyze that into 2 products. And I don't have pictures of it, and I can't really upload any photos of it, and it would take me forever to draw. But PIP2 is basically 2 parts. Part of it is an inositol group, which has had some extra phosphate groups attached to it, which is obviously going to be very soluble, and the other one that it leaves behind is diacylglycerol, which is going to stay embedded in the membrane. So just for reference, for the IP3 stands for is inositol 1,4,5, that's just giving the carbon locations, triphosphate. So basically inositol with 3 phosphate groups attached to it. And this is going to be soluble, so it's going to be floating around in the cytoplasmic spaces. It is very, very shortly lived. It's going to quickly be degraded and broken down by other enzymes. And what's most important about it, as far as the effector function of it, is whenever it binds to the smooth endoplasmic reticulum, it causes calcium ion to be released. Calcium ions are going to be released at the smooth endoplasmic reticulum. And this is going to do 2 things, right? So this is going to work with DAG, but I'm also going to mention what calcium does. Is calcium will usually form some binding with something known as calmodulin. So why would we do this? Why would we bind calcium in calmodulin, or sometimes we can leave it in the free floated form? But in the purposes of calmodulin, well, there's certain things that we need to consider. If it's bound to a protein, just think about what that's going to do. That's going to increase, in this context, the collision probability. Right? Since it's a bigger molecule now that has bound to that calcium. And another thing that it's going to do is it's going to play a role in activating calmodulin-dependent kinases. And this is abbreviated as CAMs. Now these guys are going to do other things that are going to cause a long series of phosphorylations. The book doesn't go into much detail with that, but understand that this is another mechanism. If you take an anatomy and physiology, you're familiar with hearing calmodulin. This plays a role in smooth muscle contractions. Very unique protein there. The other thing that we get from the cleavage of PIP through the action of phospholipase C is something known as diacylglycerol. Now this, however, stays bound to the membrane, so it's insoluble. Now I'll put it in green to say what DAG does. DAG is going to play a role in activating of protein kinase C. So protein kinase C activation. Now the other thing that happens though is this calcium that is released from the smooth endoplasmic reticulum. This is the free-floating calcium. This is not calcium that's bound to calmodulin. This is just free calcium cations are going to bind to and activate protein kinase C with the help of DAG. Now, this is really important because, like I said over here, that IP3 is really quickly degraded because calcium is a controlled substance for the cells, right? It doesn't want to have, you do not want to have a lot of free-floating calcium inside of our cytoplasm because that's going to do a lot of really drastic things. It can cause DNA to precipitate. It can cause uh, malfolding of our proteins, right? It can really put impeding on metabolic function. So calcium is a great cation, yeah, and it plays roles that are needed to, but it's a very controlled species. So we're not going to have a lot of calcium released, and we're not going to have a lot of IP3 bound to its receptor site at the smooth endoplasmic reticulum. That's going to be degraded really, really quickly. So one of the many functions that protein kinase C is going to have is it's going to activate this huge series of events. There's a lot of things that are activated from protein kinase C. I'm taking immunology right now, and we're also talking about things that are activated by protein kinase C. And this applies to cell biology and all sorts of things. But if you remember, there's really subclassifications of kinases. These are ones that are going to be phosphorylating serine and threonine amino acid residues. So the other thing that I wanted to mention in this context was really just the identity of a secondary messenger. How do we identify and how do we really define what a secondary messenger is as opposed to a primary messenger? Well, primary messengers are always going to be things that usually bind to receptors on the cell surface, so like proteins or hormones. That would be an example of a primary messenger, something that leads and directly acts on the cell. The secondary messengers are just that, messengers inside the cell, but what defines them is that it's usually a site of pretty intense amplification. And what I mean by that is we're being able to produce from the single primary messenger binding site or primary messenger activation. This is going to cause the production of many second messengers. So just to give you some examples of things we've already talked about. We've talked about cyclic AMP. There's also cyclic GMP. Both IP3 and DAG would be considered secondary messengers. Calcium in the solvated form and whenever it's bound to calmodulin. All of these things here, as long as once we have that initial binding here, we have this activation of this cascade, which is going to go on here. If you notice, like this here, this whole process here, really for the most part is regulated by that little timer, that intrinsic timing that we have of the GTPase activity of the alpha subunit.